Hello everyone. So today I will speak about genomics for selection and improvement of better pulp millet genotypes. So myself, Dr. Harshraj Shinde. I'm postdoctoral scholar in University of Kentucky. So currently I'm working on genomics and epigenetics and genetics group in under Department of Horticulture here. So during my PhD, I work on pulp millet. So here I will speak mainly about the salinity trait and how genomics we applied to study the salinity stress in pulp millet. So this is the outline of my talk. So in introduction, I will give about introduction about salinity stress and its impact on agriculture. Then I will speak about pulp millet. Then its abiotic stress tolerant capacity. Then I will speak about various studies which I conducted to study the sanitary stress in pulp millet. So first one is the transcriptome analysis between different pulp millet lines, that is stress, sanitary stress tolerant and susceptible lines. Then I will speak about small RNA sequencing, which we did in pulp millet to find out the microRNAs related to sanitary stress. And finally, I will speak about one of the transcription factor gene, that is NAC21. This gene we discovered in our first study, that is transcriptome analysis, and then we carried out the functional analysis of this gene in Arabidopsis, and this gene proved to be a sanitary stress tolerant gene. And then finally, I will conclude my talk. So salinity, it is mainly caused by a combination of sodium, magnesium, calcium, and other ions in the soil. So it has estimated losses in agriculture. Yield losses in agriculture due to salinity are 16%. Globally, a lot of land is affected by salinity stress. 20% of irrigated soil is affected by this stress. So in this picture also, you can see the how salinity stress has caused the soil problem and in rice. So if you go into the global picture, a lot of soil at global level has been affected by salinity stress. Right from here, Middle East area, Middle East Asia to Southeast Asia, most of the soil here is highly affected by salinity stress. Speaking about India, still the percentage is low as compared to the other Asian part. In case of Australia, a lot of soil is affected by moderate amount of saline soil. So salinity is the global problem. Then I will speak about the crop which which we studied here, which is pulp millet. The botanical name is Pennisetum glycom. In Marathi, we call it as a bajri, and in Hindi, we call it bajra. So it is staple food crop for more than 90 million people in Africa and Southeast Asia. In India, also a lot of farmers, they are dependent on pulp millet for their food and survival, especially in Rajasthan, some dry areas of Gujarat, even in Maharashtra. So it's kind of important crop. If you go the study wise, the number one is rice, number two is wheat, number three is maize, the number four is barley, and then it comes the pulp millet. So it is one of the staple food crop. It is originated in West Sahara zone, which is very hot. It is diploid. It has the diploid chromosomes with 2n is equal to 14. And in 2017, Rajiv Varshney group of ICRISAT, they sequenced the genome of pulp millet and they estimated the 38,000 genes in pulp millet. And it has very high photosynthesis capacity as compared to other crops. So The main feature of this crop is abiotic stress tolerance. It is drought tolerant crop, so it can survive at 250 mm of rainfall. It is high temperature resistant or heat resistant crop, so it can flower at 42 degrees Celsius. It is a salinity stress tolerant crop also, so it can survive up to 250 millimolar of NaCl of 250 millimolar of NaCl. So my main topic is on salinity stress here. And if you go into the nutritional value, it has very high or very good nutritional profile. And if you compare it with rice, it has very high protein, almost 1.6 times more proteins as compared to rice. High amount of fibers. It's good for, that's why it's good for digestion. High amount of minerals, and almost 30 times more higher amount of iron. So it's good for pregnant and lactating mothers. It has 4 times higher, 3 to 4 times higher calcium content. So again, it's good for infants or lactating mothers. So if you go into the nutrition value, it is highly nutritious and good nutrition profile crop. But despite of having all these characters, pearl millet is one of the least studied crop. If you go into the number of research publications, so as compared to rice, wheat, maize, or barley, or other cereals, it is one of the least studied crop. Since the genome sequencing in 2017 by ICRISAT Rajiv group, it's a research in pearl millet has been advanced. Many scientists and researchers has been attracted to this crop. So aim of our study was to study the physiological, molecular, and biochemical responses of pearl millet to salinity stress. So in case of molecular responses, we perform the transcriptome analysis. Very few biochemical responses we studied. Physiological responses of different pearl millet genotypes were also studied. The genes which we got in transcriptome analysis, that is in RNA sequencing, which studied one gene. Another students in Japan, they are studying now another genes. Then functional analysis of such stress tolerant genes. So I will start with my study one. First one is the comparative de novo transcriptomic profiling of the salinity stress responsiveness in contrasting pearl millet lines. Here we perform the RNA sequencing of different pearl millet lines. So material and methods or seed materials of pearl millet. So initially in 2016 we got the seeds of pearl millet genotypes from ICRISAT. So especially pearl millet breeding station, Dr. Shashi Kumar Gupta, they provided us the pearl millet seeds for this study. So they provided 8 lines, 4 were salinity tolerant lines and 4 were salinity susceptible lines. Then we conducted some of the trials in greenhouse for salinity stress tolerance and susceptibility to have the perfect lines for our future RNA sequencing experiment or transcriptome studies. So we, based on our initial screening in greenhouse, we selected ICMB 01222 as a baseline or perfect salinity tolerant line. And at the same time, we selected the ICMB 081 as a perfect susceptible line to study the contrasting nature or contrasting phenotype during salinity stress. So after selecting these 2 lines from our initial trials, so we again perform the physiological analysis of these 2 lines to compare them. So we grow both these lines in same pot. So here you can see that this left panel is under control situation. So you can see under control situation, both these lines has almost no effect. They are growing healthy, but after like 16 days when we applied stress, salinity stress, By giving them the 250 millimolar of NaCl through irrigation channel, you can see the clear difference. You can see the salinity tolerant line is still healthy, almost no effect on their leaf, no effect on their height, little effect on their height. But on other hand, if you see in the salinity susceptible line, that is ICMB081, the leaf started drying, lots of symptoms like deep burning, loss of chloroform, yellowing we observe. So we, based on this analysis, we think that both are the perfect tolerant and susceptible lines for our future studies. Then we perform some of the biochemical responses of physiological responses of polymillet to salinity stress. So we measured the relative water content, and you can see that during stress, tolerant line had higher relative water content as compared to susceptible line during stress situation. Although the relative water content was decreased in tolerant line, but it was decrease was very high in susceptible line. At the same time, we calculated the chlorophyll content by SPAD meters, and we found that chlorophyll content was very less in case of susceptible line during stress. And in previous picture also, you could have seen that they almost showed the yellowing, that is almost there was the loss of chlorophyll. But on the other hand, tolerant line had still higher chlorophyll content than susceptible line. And this was our key finding about the sugar. During stress situation, the sugar was increased in both genotypes, in tolerant and susceptible. We think that sugar is the key here. Tolerant line showed almost 1.6-fold higher amount of sugar during stress. In case of susceptible line also, sugar increase, but the amount increase here was very less as compared to the tolerant line. And then same again, during stress, higher the sodium accumulation in leaf, the less susceptibility of that line. So you can see the susceptible line has accumulated high amount of sodium in leaf. And we also observed sodium injury symptoms, and so some of the sodium injury symptoms like leaf tip drying, leaf yellowing, and stunting were observed only in susceptible line. Then based on some of the physiological analysis and biochemical analysis like relative water content, chlorophyll content, sugar content, and some of our phenotypic observation, we selected these two lines and we performed the RNA sequencing of both these lines, and we maintained the 3 replications. We selected the leaf samples from both control and salinity stress plants, and we selected these and we sequenced them using Illumina HiSeq platform. After sequencing, we got raw reads, then we filtered the raw reads into clean reads. We checked and we converted them into the contigs to find out the genes, to find out the genes. And after finding the genes, we performed the differential gene expression analysis. Of course, here our motto was to find out the genes related to salinity stress in both lines. So we performed the differential gene expression in R using edge test, and around 2,900 genes were upregulated. The same amount of genes were downregulated in tolerant line, that is ICM0122. But in case of susceptible line, less number of genes were upregulated and higher number of genes were downregulated. So we think that this is the key difference. The more the upregulation of genes, the more or better response of these genotypes. So here you can see that there was less amount of upregulation in susceptible lines. Then we draw some of the common genes between these two genotypes. So these are the commonly upregulated genes and common downregulated genes between these two genotypes. So after finding so many genes in both genotypes, we went for annotation of genes to find out the specific gene families. So we started with NAC transcription factors. Sorry, we started with transcription factor families identification. A lot of families of transcription factors we identified, but here we have shown only top 10-15 families here. So most uh, the the family which we observed most in the we involved in sanitary trace of palmillet was jing finger. This was the uh, this family has the most number in palmillet during sanitary trace in both tolerant and tolerant and susceptible. Other than that, we found some of the other families also, that is APT transcription factors, MIB transcription factor, NAC transcription factor, Werke transcription factor, and W8 transcription factor. And there were lots of other transcription factor families also we observed. But the key difference between tolerant and susceptible lines we observed was squamous of promoter binding transcription factor, because this transcription factor family shows expression only in tolerant line ICMB0122. Here we have also highlighted this family because this family was only observed in ICMB0122. So future studies are needed to study this family in detail. After finding the transcription factors, we went for ion transporters identifications, because during salinity stress, ion transporters plays key role in ion detoxification. So we found around 6 families of transcription factors. First one is ABC transcription factors, which ATP binding acids and high efficiency potassium transporters, sodium hydrogen exchange and other transporters also. So here also, if you see, this transcription factor shows very high upregulations or very high expression in tolerant line as compared to susceptible lines. So here again, I would like to emphasize that more the expression of transcription factor, the better tolerance of that genotypes. So here you can see almost all families showed very high enrichment in tolerant line ICMB0122. Then we perform the cake pathway analysis to find out the exact metabolic pathways involved in salinity stress on pearl millet. We found 300 pathways, but it was difficult to show all 300 pathways here in our analysis. So here we have shown only 20 best pathways or 20 most significant pathways. So different pathways, here pathways showed by different colors for susceptible, tolerant lines, the down-regulated and up-regulated pathway. This panel is down-regulated pathways and this panel is up-regulated pathway. So among pathway, the key difference was the glycolysis and ribosomes, then phenylpropanide and ubiquitin-mediated proteolysis pathways, because these pathways shows very high. For example, glycolysis and gluconeogenesis, this pathway shows very high amount, very high down regulation in susceptible genotypes, but this pathway were almost showed very less down regulation in tolerant genotypes. So this pathway were also a key pathway. And on other hand, the phenylpropanol biosynthesis and ubiquitin mediated proteolysis, this pathway shows very significant upregulation in tolerant genotype as compared to susceptible genotype. So we think that this pathway also holds key for stress tolerance and susceptibility during salinity stress. So key finding from our this study was the tolerant genotype ICMB0122 showed higher growth rate and accumulated high amount of sugar in leaves under salinity stress. Squamous promoter binding transcription factors were only found in saline tolerant line. Ubiquitin mediated proteolysis and phenylpropanol biosynthesis were upregulated in the tolerant line. And we think that tolerant line can be used as a donor lines for the donor, as use can be used as a donor of useful genes to further improve the salinity tolerance in pulp millet. So our next study is the small RNA sequencing to find out the microRNAs related to salinity stress. So here, based on our previous studies, we selected only tolerant genotype. We got some of the raw reads and clean reads. After sequencing, then we went for differential expression of microRNAs. So in this study, we found 30 upregulated and 51 downregulated microRNAs. Among these microRNAs, we identified the microRNA 159 was the most significant family in case of silent stress. Then we went for the target gene identification of for these microRNAs, because microRNA depends on their target genes for their functioning. We identified different families of target genes. Among them, squamous promoter binding and MIP transcription factors were the most enriched target gene families. Then we also went to find out the pathways which microRNAs regulates during salinity stress, and we found that during salinity stress in pulp millet, microRNAs and their target genes regulate the oxygen signaling pathway. Here you can see that this upregulated and downregulated microRNAs regulates many oxygen signaling pathways genes. And these genes finally goes for salinity stress response during salinity stress. Then we also validated the expression of these microRNAs by qRT-PCR. Almost by in our qRT-PCR result, microRNAs showed the almost similar expression pattern like RNA sequencing. Then we also performed the qRT-PCR analysis of microRNAs and their target genes at the same time. These are the microRNAs and these are their target genes. And in our analysis, we found that both these microRNAs and their target genes shows contrasting expression patterns which we expected. So key findings of our studies are 81 microRNAs we discovered in pulp millet which are salinity stress responsive, and we identified 448 target genes for these microRNAs. Around 25% of them are transcription factors, and these microRNAs regulates oxygen responsive pathway during salinity stress. And we think that some microRNAs plays role in salinity stress tolerance mechanism of pulp millet as like other crops like Arabidopsis and rice. Then our final study, we studied the NAC transcription factor of pulp millet, that is PG-NAC21, and we found that this NAC transcription factor enhances the salinity stress tolerance in Arabidopsis. So this NAC21 we found in our RNA sequencing analysis. So NAC are important family genes, and here we perform the functional studies of this gene, NAC21 gene. We perform the gene expression analysis of NAC21 during salinity stress, and you can see as time of salinity stress goes, after 6 hours of salinity stress, it was upregulated. After 12 hours, it was still high upregulated, but during 18 hours of salinity stress, it shows almost 24 to 5-fold change. So based on our RT-PCR analysis, we think that this gene is best candidate gene for future studies. And we also found that this gene's expression is also upregulated by ABA treatment. ABA is the abscisic acid and stress hormone. So that's why we selected the expression, we selected ABA for our analysis. Then we constructed the transgenic lines of Arabidopsis, which shows the transgenic lines of Arabidopsis, which overexpresses the pulp millet NAC21. And then we perform the salinity stress tolerance assay of this transgenic line. Here This is the control condition. You can see all plants are growing well. Col 0 is the wild type. This is the transgenic plant line 2, transgenic line 6, and transgenic line 3. So here in this picture, you can see as compared to wild type, the transgenic lines are showing high amount of germination. Then their fresh weight is very high. So based on our these studies, we think that NAC21 is the best candidate genes to study the sanitary stress in pulp millet. So this is one of the key candidate gene. Then for same NAC21 overexpression lines, we perform the root assay. So you can see during the salinity stress, overexpression line showed the very high amount of root elongation as compared to the wild type lines. So we think that this gene is also responsible for root elongation during salinity stress. Then we find out some of the target genes of NAC21 in Arabidopsis. Most of these genes are stress responsive genes. And then we propose the model how NAC21 works in pulp millet. So key findings of our studies are expression of NAC21 is upregulated by stress, salinity stress, and ABA. MIB transcription factor bind to the promoter of PGNAC21. These results we have not shown here. And I have shown the result of transgenic lines. So transgenic lines of Arabidopsis, those who overexpress the pulp millet NAC21 has exhibited the stronger tolerance to salinity stress than the wild types. PGNAC21 overexpression Arabidopsis plants also has shown the enhanced expression of stress responsive genes. So finally, conclusion from our all three studies. Understanding the effect of salinity stress on pulp millet and their mechanism of salinity stress tolerance are vital for breeding new salinity stress tolerant cultivars. Based on our RNA sequencing study, we found that the total soluble sugar content was higher in salinity stress tolerant pulp millet lines, and sugar metabolism related pathways were highly upregulated in the tolerant line. So we think that the sugar is the key. Higher the sugar, higher the salinity stress tolerance in pulp millet. MicroRNAs also mediate the stress tolerance in stress response in pulp millet. So, and we found that they regulate the stress response in pulp millet through oxygen responsive pathway. NAC21, one of the genes which we studied, is also the key stress responsive gene and is the positive regulator of salinity stress tolerance. So finally, I would like to acknowledge ICRISAT, International Crop Research Institute for Semi-Arid Tropics, India, for providing the pulp millet genotypes. And also special thanks to Dr. Shashi Kumar Gupta for their guidance for pulp millet different assays. We would like to acknowledge Nodai Genome Research Center of Japan. They funded us for RNA sequencing analysis. Finally, I would like to thank the University of Tokyo, Japan, where I did my PhD and where all this work I carried out. And also thanks to University of Tokyo for providing Todai special scholarship to me. So based on our all three studies, we have published 3 papers. So these are the titles of all these 3 papers. First one is for RNA sequencing, second one is the microRNAs, that is small RNA sequencing, and third one is NAC transcription NAC21 studies in pulp millet. So among these papers, microRNA papers has been published recently last month. So if you have comments, questions or suggestions for our future directions, these are my email ID. You can send me emails for any kind of suggestion, comments or questions. Thank you for attention. I hope you enjoy my talk. Thank you.